So imagine if all of the music on iTunes was interrupted by blocks of white noise that you had to manually edit out before every time you played a song. Makes no sense, right? Why would they be recorded that way? Why not just remove the junk at the source so you don't have to waste the time and energy? That's exactly what scientists wondered when they discovered genetic material known as introns. Our genes are like molecular messages, and those messages are interrupted by segments of DNA called introns. You can think about all the DNA in your bodies as a massive library of music. The genes are like songs that can play at different volumes, and the introns are the white noise. Introns were originally called junk DNA because they're actually removed from genes when molecular messages are processed. So like the white noise, they're not present in the final product. So they have no effect on the trait encoded by the gene. Yet over 25% of the DNA in your cells is made up of introns. So why do our cells waste energy and resources maintaining and copying these introns if they're just going to be cut out every time a gene gets turned on? Let's return to the iTunes analogy. Imagine that iTunes gives in to customer complaints and they decide to get rid of all the white noise. You're surprised to find that suddenly your songs are playing at different volumes. Some of them are playing much more quietly than they were before. And some of them you can't really hear at all. That's essentially what happens when scientists try to study genes without introns. Without their introns, some genes are played very quietly, and some never turn on at all. Now, even though introns act as a volume control for some of our most important genes, we actually know very little about how this process works. In my lab at UC Davis, we study this phenomenon in plants because introns are routinely used in plants to turn up the volume of useful genes in genetically engineered plants. For my thesis research, I am working to better understand this mechanism so that we can unlock the potential of introns for fine-tuning the volume of genes. Introns have already been used to boost vitamin content in nutritionally enhanced rice, to improve yields in soybean for farmers, and more. If we can unlock the key to their mechanism, we can specifically design introns to orchestrate a symphony of genes with useful purposes. In the future, introns might help us increase the productivity of plants for use in biofuels, or even enable plants to produce life-saving vaccines. Thank you.